So thanks to New Phytologist for the invitation to speak at the symposium. The results I'm going to present today are due to hard work by a very large number of team members on the 1KP project, which is what a lot of my talk is about. 1KP is a large-scale comparative transcriptomic initiative that is funded by the government of Alberta, province of Alberta, and BGI, Beijing Genomics Institute, which is in Shenzhen, initiated and led by Gain Wang at the University of Alberta. So 1KP is beginning to have a very far-reaching effect in different fields of biology, including many studies of gene family evolution that might be of interest to people here that I'm not going to talk about today. The first of a series of major capstone papers to the project is nearing submission and will be coming out shortly. This figure shows part of a large phylogenetic tree for the slice that you're seeing here is for ferns and relatives. The width of the lines in this figure is proportional to gene diversity, or the depth of gene diversity for these lineages. That single very thick line you can see on the left is the Phylogeny genome, and you can barely see the other lines because they're barely there. That's what's on GenBank for ferns and lycophytes, really a few genes, if we're lucky, for individual species. On the right is the amount of data that is coming from 1KP transcriptomes. Same phylogeny. Some of the major groups here include the manilophytes, leptosporangiate ferns, and lycophytes. So a much denser sampling of gene diversity. The taxa that have been included in 1KP, the name comes from 1,000 plant transcriptomes, have been chosen to represent phylogenetic diversity, taxonomic diversity well across all of the green plants, including all of the major land plant lineages. So although they're not visible here, we have a real nice diversity of ferns in the sampling. The tissues that we used it's not really possible to find a consistent tissue across these taxa, but we use sort of green shoots or something like that in most of these cases. This is the whole tree, and as you can see, most of the gene diversity that was sampled is present in the angiosperms, and that's just a function of most of the species are in angiosperms. But in fact, we oversampled in the non-flowering plant land plants. So all of the land plants outside the angiosperms, gymnosperms, ferns, lycophytes, and the 3 bryophyte lineages. Although these seem a bit thin compared to the flowering plants, they're actually sampled in some cases at a much higher density than clades within the flowering plants. And we did that on purpose because I was in control of the sampling of this part of the tree. Before I really get into the results, I want to thank Jim Liebensmack and his crew of people at the University of Georgia. So they supplied the technological know-how to analyze the data. A lot of the analyses, if you're interested in how the data were produced for what I'm going to discuss, basically followed this capstone paper, or this pilot paper, sorry, that came out in 2014 with a subsampling of taxa, about 1/10 of the total data. So the methods that we use are basically the same with a few tweaks. Most of the trees that I'm going to show are based on an effort to recover single copy or close to single copy genes from this sampling, because these are the easiest ones to deal with, for now at least, in phylogenetic inference. So one of our major goals is just to prove that we can do this, and that these genes collectively have useful phylogenetic information. I was in charge of this slice between the flowering plants and the algae with Dennis Stevenson, but really it's thanks to the efforts of more than 100 sample providers who really went out of their way to contribute to the project that this was even possible. in an audience like this, I probably don't need to say this, but this audience is actually a little bit more diverse than what I'm used to talking to. So because my talk has a lot of phylogenetic tree information, sometimes I feel like I have to justify it a little bit. And it's always, when you're wanting to justify something in biology, to turn back to Darwin and to see what he had to say. So he had lots to say about this. He thought phylogenies were really important. It's sort of a bittersweet quote. It's actually a slice of a letter he wrote to Huxley, in that he could foresee the time when phylogenies would become easily producible and available. In fact, we now live in that time. So we live in sort of the golden age of systematics, because we can do this. And having these phylogenies is useful just in and of itself, but we can make use of them to think about analogy, what we now call homoplasy, and homology of characters. So Darwin, no doubt, would have been deliriously happy to see something like this. This is a summary phylogeny of land plants. And I'm showing here the major groups of land plants. So I just condensed that little angiosperm group to one line there, but I think some of these other groups are, in their own way, at least as important. The mosses and ferns are the two most numerous in terms of species groups outside the flowering plants, but there are other important lineages here too. And so if we have a phylogeny like this, we can potentially use it to think about trait evolution on a tree. So there's some very simple things you can do in this one. Invasion of the land is sort of a no-brainer, and vascular system defines vascular plants, a seed defines seed plants, and so on. Of course, sometimes these simple-minded mappings break down, so that mapping of true leaves, which gives name to this clade, actually, the euphylophytes, may not be quite right, because we don't really understand how many times leaves have evolved, or what the true homologies are in leaves. And of course, leaves also evolved here in the lycophytes. So we can use these phylogenies to think about things like leaves, roots, stomata, and so on. For somebody training a new generation of plant systematists, it's actually a bit of a relief to have things to work on. So these are slices of that same land plant tree highlighted in red, which really signify parts of the tree where we don't understand relationships. So some really critical parts of the tree we don't understand. What the first divisions are in the land plants, for example. We don't understand relationships within the gymnosperms, within the seed plants. We don't know what the sister group of the angiosperms is, really. It's pretty astounding. We don't understand relationships among the major groups of ferns, and so on. And pretty much every group that you could look at here, you can pick out other examples of things that we don't know. Within mosses, what's the sister group of the rest of the mosses? That's not clear. So part of what we want to do is to see if we throw a lot of data at this problem, we can resolve some of these relationships. This is a figure from the pilot paper, where we summarize some of the major controversies in land plant phylogeny. And I'm just going to focus for now on one of these that involves the very first splits in the land plants. So you're looking at three representatives of the three bryophyte clades, the hornworts, mosses, and liverworts. And of course, the fourth major lineage in land plants is the vascular plants. You can pretty much go into the literature and find any set of relationships that you want for these. I think pretty much everyone is out there somewhere. This is 5 that we selected just to show in the paper. It's worth remembering that many of those relationships are based on early phylogenies with poor samplings that were possible at the time. They're weakly supported and so on. So it's perhaps not too surprising that there is this diversity out there. So in this pilot study, led by Norm Wickett at Chicago Botanical Garden, we took a couple of major approaches to taking these several hundred single copy nuclear genes and analyzing them. And you really have, apart from choices to make with decisions to make about whether to analyze it at the DNA level, or the amino acid level, or codon level, you have a major choice to make about how to deal with the different genes. So you can just combine them into one single concatenated matrix. It's one very popular approach and very effective approach often. Or you can worry about inconsistencies that there might be between individual gene trees. And one major source of those inconsistencies is this process called incomplete lineage sorting, where we can have temporary retention of polymorphisms between successive speciation events. And that's a problem for phylogenetic inference because it can lead to the gene tree being correct as a gene tree, but not really matching what a containing species tree might be. And so there are approaches out there that actually try to use these inconsistencies among trees to estimate a better species tree. So Astral is the sort of current most popular way to do this. And we used both approaches and many different sort of iterations of models and so on to get a grip on this. The slice I'm showing is at the base of the land plants. You can maybe see hornworts, liverworts, and mosses, and so on. So I simplified it here to show the results. And as you can see, we inferred relationships really confidently twice. So these are conflicting relationships, right? And the numbers on here, the closer they are to 100%, the more confidence we should have in them, except they tell very different stories. So there's something worrying and there's something going on there. You can look in the literature. There are some other phylogenies that have good support, and they tend to have this topology. So this is some work that a PhD student of mine, Ying, who's in the room, produced a few years back with a nicely resolved phylogeny of land plants, very nice support. Here's a study by Yinglong Chu, who also found a similar phylogeny with different types of data, but both are sort of heavy with plastid information. These are the same topology, right? But these are all different. So for example, liverworts and mosses are closest relatives here, and they're a grid here. Hornworts are sister to vascular plants here, and they're sister either to the rest of the land plants or to this group of bryophytes here. So really, these are really different trees, and they have very different implications for character evolution. So which is correct? Well, maybe they all are. So if you were to deroot these trees, as far as I know, these are the only well-supported tree shapes that are out there. So if you deroot them, they're basically all the same tree. This is an unrooted 4-taxon tree, and you can put a root in different places and come up with those 3 trees. So I think that's pretty much as far as we got in this question. The capstone analysis is running into the same problems with astral versus concatenated analysis. So I don't think it's going to go away immediately. I think what we have here is a sort of classic long branch attraction problem. So that's a problem where we have a short intervening branch that separates major groups that might be on long branches, where there might be a long outgroup, and there definitely is in this case. There's a very long gap between the land plants and the closest green algal relatives in terms of time. This is an overview of the capstone analysis, and the relevant part of that 4-taxon tree is actually hidden under that dot. So it's a very short branch, and this is the branch connecting land plants to green algae. There's branches to hornworts and liverworts and so on. So it's probably a long branch problem, and it's probably a situation where slight differences in the model or different methods are going to nudge it particular ways, maybe none of which are particularly believable. So now on to some good news. I'm going to spend the rest of my talk talking about 2 groups of land plants, mosses and ferns, a little bit shorter time spent on mosses, just to show you that actually we can do a really good job with these kinds of data. So here are a couple of examples of uncertain relationships within moss phylogeny. So everyone's heard of sphagnum. Probably most people have not heard of Tachycea, the impossible moss that was first thought to be a liverwort. It has kind of a liverwort look to it. So some recent phylogenies have actually put this as the sister to the other mosses rather than this. And then within each of the subclasses of mosses, there are uncertainties. This is the biggest subclass of mosses by far, or this is the biggest class of mosses by far with 6 subclasses. And the relationships among these subclasses are not all certain. This is Fiscomatrella, by the way. This is Buxbaumia, which one of my students, David Bell, has a poster on that I'll be talking about later. So just to cut to the chase, this is the backbone that is inferred in the unpublished capstone analysis. And I'm not showing all the relationships on here. I'm just showing how the different groups are related to each other, because that's of interest to me, the relationships among those groups. A star here means 100% bootstrap support, both from the concatenated analysis and from the astral analysis. This has been done in different ways. This particular version is an amino acid DNA substitution model, but they all pretty much have this sort of same sets of relationships. So pretty much there's a lot of confidence here, if you like, in relationships. This particular one has proved to be a very hard one to resolve between whether there's a clade of tetraphytopsida, polytrichopsida, or whether they form a grade and so on. We don't have a few lineages samples, so Odipodium and Androbrium have not been sampled yet. They're hard to get hold of, but it would be nice to slot those in there. So that's, you can take a data set, perform a bootstrap analysis on it, or a posterior probability analysis, Bayesian analysis, and you can come up with good support values. But for me, the proof of the pudding for any phylogenetic study is to compare it to other studies and see if we resolve comparable sets of relationships. So this is a phylogeny produced a couple of years ago by Ying Chang in my lab. Ying is also here, based on a bunch of plastid genes. That's a pre-next generation data set, but a very good data set. And you probably can't see much on here, but I'll just tell you that it's basically the same backbone that you saw previously. So that's something that's of great comfort to plant systematists, or to systematists in general, when you have this congruence of results. Another student in my lab has been looking at mitochondrial genes. You can pull them out of 1kp data. And so he's been able to basically get full mitochondrial genome gene sets from a lot of the taxa, and we've supplemented that with genome skims that we've done in the lab and a few things from GenBank. And basically, again, it's the same story. Almost all of the relationships are the same. Where they differ a little bit is just a weakness in that particular tree. So I think we're zeroing in a really confident set of relationships for mosses. Takaki is probably sister to the other mosses. Some of the relationships among the subclasses are becoming really clear. This was meant to be my light relief. Does everyone know who this is or what this is? Some people are nodding. So this is Zach Galifianakis. You can find these skits or sketches online. He interviews Obama, you name it, in between two ferns. So that's the name of the sketch. Does anyone know what kind of fern this is? Yeah, well done. So Emily Sessa, I think this is the best title of a paper I've seen in the past few years, Between Two Fern Genomes. This is a position paper saying it's an absolute absurdity that we don't have any sequenced fern genomes. And so we're proposing to do that here. I think we should get a whole lot of them because they form a critical position in land plant phylogeny, and it hasn't been done. Fern people don't think about their ferns, they should. Botanists definitely should. So here's a summary of major groups within the group that's now being called ferns broadly, sometimes called the manilophytes. Some people like those names, some people don't. But the classic ferns are things like the leptosporangiate ferns. Here's a leptosporangium, or meradioid ferns, ophioglossoid ferns. People get a little bit less comfortable when you start talking about horsetails and whisk ferns as true ferns, but they're in this group, so it's just a name. Names aside, we don't have a very good idea of relationships among these major groups of very morphologically diverse organisms. And again, the Capstone analysis is unpublished analysis with the same kinds of analyses done, gives a very strongly supported set of relationships amongst the five major groups of ferns, and within the biggest group, the leptosporangiate ferns, where most fern diversity is. Again, we have a very good sense now of these backbones of relationship, which has also come from other studies, but I think 1KP will hit this on the head. So most biologists probably, this is how you look at next generation, sort of a big box, and you throw your data in and you get hopefully something nice out of it at the other end. It's sort of how I view it. I worry about that a lot because those of us who grew up doing manual sequencing and dealing with sequence files and so on know the sorts of things that can go wrong, and it's often only by sort of supervising the process of putting data sets together that you figure out that there's a problem. So data sets are now too huge to do that, and so we have machine creation of data, and that worries me a bit. It worried a colleague of mine, Carl Rothfels, and he and a couple of colleagues took it upon ourselves to go back into the 1KP data and to pick out sort of like a hand-curated, a boutique version of transcriptomics. I guess my sort of, I'm going to throw this at Carl the next time I see him, but I think it's sort of hipster transcriptomics. That's what I'm going to call it. Where you're kind of like, you're trying to do everything to the finest possible way. So Carl actually went in and he manually pulled these sequences out and put it with some machine assistance, but supervised every step to generate an alignment of 25 genes. And they did some really careful phylogenetic analyses with a broad range of models and methods. And basically whatever he threw at it, he got this result. And it's also basically what the Capstone, the full set of 400 genes produces. So this again is sort of confidence building that we're zeroing in on the right answer. And this is just to summarize the Capstone and Carl's Hipster method that basically find the same sets of answers with strong support. So I thought I'd finish off with, I love phylogenies and I love seeing them being produced and thinking about things like congruence and different methods and so on, but I think that can be a bit dry for general audiences. So I thought I'd pick a topic that is probably appealing to most people here. This idea of how we place fossil plants in phylogeny, and of course I'm not a paleobotanist by a very, very long shot, but I am interested in plant form, and I worry about these. So this is from a couple of papers by Gar Rothwell and colleagues. Basically, sort of being Gar can be sort of the cranky old guy. Of anyone who knows him, that's who he is. But he shakes his fist sometimes at people doing molecular systematics, and you guys don't understand this, and you're doing it wrong. And he's probably right. But he worries a lot about this, and So this particular set of papers were about where ferns place in plant phylogenies. So it turns out that there are probably major radiations of ferns that happen both within the clade that we now call ferns or manilophytes. So he calls them first, second, and third radiations. But there are probably other radiations of things that don't belong in that clade that fit somewhere along the stem leading to the ferns, or maybe to the euphylophytes, the ferns plus seed plants. So things like these maniliform ferns like cladoxylon, steropterids, and zygopterids. I'm going to express the frustration I have with ferns when I teach. And it sort of is relevant to this question of what is a fern. So it's rarely actually written down on paper. You can look up a textbook what a fern is. The best definition I can come up with, and it's the one that I teach with, is that a fern is a vascular plant that has megaphyll leaves with sporangia on the leaves. Sporangia borne on the leaves. That is not a seed plant. So you have to include that in there, even though seed plants have sporangia on things that might be leaves. But so it's sort of a very kind of loosey-goosey definition of what a fern is. And I think that's partly part of the problem, that we try and think about ferns as this one set of things, where it's just a sort of whole smear of things that have probably evolved repeatedly in different lineages. So Gar took an analysis that he had done a couple of decades ago, building up a large morphological data set of ferns that included extinct, these X's here, and extant lineages, and did some phylogenetic inferences using parsimony on these, with these number of characters and number of taxa, not a good ratio to have here. And surprise, surprise, if you try to do bootstrap analyses on these sorts of data sets, you get basically no support for the backbone. So for anyone who wants to, I don't know, if I was wanting to publish this, I would be really uncomfortable, but I don't have a good sense of how well supported these relationships are, but he published it. He also reanalyzed a data set by Pryor et al., which was a data set of morphological data set for extant taxa, where he added extinct taxa. And the result was even more discouraging because the backbone is basically collapsed. There's nothing to that. So what do you do in this situation? Well, what I did this weekend was I thought, well, why don't we take the 1K piece analysis and just see, use that as inspiration and see where the morphological placements of these things may be if we try to use this as a sort of topological constraint, a backbone, and see if we use a morphological data set where individual taxa would place on that. So this is an approach that's been used by other people. So Jim Doyle uses this commonly for angiosperm paleobotany. And that basic idea of using a backbone phylogeny for scanty data is actually a pretty widespread one across biology. So anyone who does environmental samples with, for biologists, for example, there are comparable algorithms for people who work in viruses to place these very skimpy sets of data on trees. So I did that using the modified Pryor et al. Data set just to see where these things would go. And many of the answers were surprisingly good, so where we would sort of expect them to go based on the literature. So seed ferns, which are seed plants that look like ferns, place here. Aglaophyton, a stem vascular plant places here. Myraidoid fern here and a water fern here. So these are sort of where we think they should go. These we sort of think are sort of early leptosporangiate ferns, so they're sort of, that looks good. Other things maybe not so much. So this is this maniliform fern that's supposed to be outside the manilophytes, cladoxylon, but here it goes in with Myraidiaceae, which is kind of unusual. Psilophyton is a trimerophyte, which should really be here. And then there's a bunch of things that really, they just go everywhere. So I think the take-home message of this is that even morphological data sets that look pretty sketchy, there is information in them, and we can maybe come up with a way of blending genomic and fossil methods to come up with some better answers. But I think this also underlines that next generation paleobotanists have their work cut out for them. So I just want to close off with sort of an announcement of a new endeavor that you might have heard of. So Emily Sessa and colleagues are leading this project to sequence 500 loci for all 18,000 species of non-flowering land plants. They call them the flagellate plants. It's actually not a good name because there are non-flowering land plants that don't have flagellate, but it's okay. So what they're looking for is partners. So if you would like to be a partner in this work, the way that many people were partners and got authorship on many 1KP papers have done, get in contact with Emily because they're sort of getting going in that. I think this is going to be sort of the next generation of genomics for early land plants. So thank you.